Hello everyone, I am Zig Villano, the representative for Group 1. Today we are going to discuss the ongoing research for the COVID-19 vaccine. Firstly, let me present the question, why are there so many vaccines in development? To answer this, we must first understand the different approaches into making the vaccine. Now let us move on to the different approaches in making the vaccine. As of now, it has been known that there are three approaches to making the vaccine, namely the whole microbe approach, the subunit approach, and the genetic approach. When it comes to designing the vaccine, their differences lie in whether they use a whole virus or bacterium, just the part of the virus that triggers the immune system, or the genetic material that provides the instructions for making specific proteins and not the whole virus. We will discuss all of these, but first let us explain the whole microbe approach so we don't get confused. The whole microbe approach is divided into three different vaccines, namely the inactivated vaccine, the live attenuated vaccine, and the viral vector vaccine. Let us discuss first the inactivated vaccine. The inactivated vaccine has a process of taking the disease-carrying virus or bacterium or one very similar to it and inactivate it or kill it using chemicals, heat, or radiation. This approach uses technology that has been proven to work for people. This is the way that flu and polio vaccines are made, and vaccines can be manufactured on a reasonable scale. However, it requires special laboratory facilities to grow the virus or bacterium safely. It can have a relatively long production time and it will likely require 2 to 3 doses to be administered. Next, we have the live attenuated vaccine. A live attenuated vaccine uses a living but weakened version of the virus, or one very similar. The measles, mumps, and rubella vaccine are examples of this type of vaccine. This approach uses similar technology to the inactivated vaccine and can be manufactured at a scale. However, vaccines like this may not be suitable for people with compromised immune systems. Next, we have the viral vector vaccine. This type of vaccines uses a safe virus to deliver specific subparts called proteins. These proteins are part of the germ, so it can trigger an immune response without causing the disease. To do this, the instructions for making particular parts of the pathogen of interest are inserted into a safe virus. The safe virus then serves as a platform or a vector to deliver the protein into the body. The protein then triggers the immune system as to response. An example of this vaccine is the Ebola vaccine. This type of vaccine can be developed rapidly. Next, we have the subunit approach. A subunit vaccine is the one that only uses very specific parts of a virus or bacterium that the immune system needs to recognize. It doesn't contain the whole microbe or use of a safe virus as a vector. The subunits may be proteins or sugars. Most of the vaccines on the childhood schedule are subunit vaccines, protecting people from diseases such as the whooping cough, tetanus, diphtheria, and meningococcal meningitis. Now we have the genetic approach. Unlike vaccine approaches that use either a weakened or dead whole microbe or parts of one, a nucleic acid vaccine just uses a section of genetic material that provides the instructions for specific proteins, not the whole microbe. DNA and RNA are the instructions our cells use to make the proteins. In our cells, DNA is the first turned into the messenger RNA, which is then used as a blueprint to make specific proteins. A nucleic acid vaccine delivers a specific set of instructions to our cells, either as DNA or mRNA, for them to make the specific protein that we want our immune system to recognize and respond to. The nucleic acid approach is the new way of developing vaccines. Before the COVID-19 pandemic, none had yet been through the full approvals process for use in humans. Though some DNA vaccines, including particular cancers, were undergoing human trials. Because of the pandemic, research in this area has progressed very fast and some mRNA vaccines for COVID-19 are getting emergency authorizations, which means they can now be given to people beyond using them only in clinical trials. Now let us circle back to the question, why are there so many vaccines in development? Typically, many vaccine candidates are evaluated before any are found to be safe and effective. For example, of all the vaccines that are studied in a lab and in laboratory animals, roughly 7 out of every 100 will be considered good enough to move into human trials. Of the vaccines that do make it into clinical trials, just 1 in 5 are successful. Having lots of different vaccines in development increases the chances that there will be one or more successful vaccine that will be shown to be safe and efficacious for the intended prioritized populations. Now you know what it takes to create these vaccines. It took researchers 11 months to give the first ever jab to a human trial. Our goal in this presentation is to acknowledge the innovative minds that made these vaccines successful and effective. Thank them for risking their very lives to save the people that needs help the most. We are Group 1. Thank you for listening.